This project, we're looking at mosses, their abundance and distribution. So we've got our transect here. And we're going to look at how to measure percentage cover. We've got a lot of sphagnum moss here, a big root, and so you're literally going to use one cell which makes up 1% of the quadrat and use it to measure its abundance within that quadrant.
Yep, and our transect line is 10 metres long and we're positioning them with 1 metre in between them. Shall we go to the next one? Yeah. Anyway, so in this quadrat, for example, of this type of moss, we'd have about 6 to 8% for this one, because there's nothing else in the quadrat. The rest would just be bare ground. And then we've got our sample pots, which are full of all the different ones that we'll ID later, as you can see here. For each of our transect line, we've drawn a little map to show where trees and shrubs would be along the transect in case any of our data is a bit unusual and we don't understand.
This is our raw data table, so we're just going to use this to measure coverage. So the species at the moment are just called A, B, C, D and E. We'll get the Latin and proper names later on. And within each of our 3 areas, we're going to have 5 different quadrats and 1 metre apart, and we'll measure our coverage of each of the species there, and we'll also account for plants and bare ground so we can see what actually is around the moss.